Hi, my name is David Thomas, and I'm the manager of the educational labs here in the Crocker Science Center. Before you get started in your lab course this semester, let's cover a few items that you need to be aware of to maintain a clean, efficient, and safe work area. First, let's talk about PPE, or personal protective equipment, beginning with the clothes you wear to lab. Closed-toed shoes are the minimum required footwear for working in your physics lab this semester. However, in any experiment where knives will be used, fully closed shoes, not just closed-toed, are required. Throughout this lab course, gloves will be available for certain experiments. They are on your hands to keep messy substances in the lab from getting onto you. You must be thoughtful when using gloves about what you're touching. Rubbing your face because of an itch, pushing back your hair, putting your hands in your pants pockets, or handling personal items with your gloves on are all ways that substances on your gloves can then be transferred to you. How you remove your gloves also matters. To remove gloves properly, grasp one glove near the palm of the hand using the other gloved hand. Pinch and pull the first glove off. This should invert the glove as it's removed so that the outside of the glove is now inside. Ball up this removed glove in the remaining gloved hand. Slide your ungloved finger beneath the cuff of the glove, avoiding touching the exterior, and then pull that glove off. Again, this should invert the glove. You'll end up with a ball of gloves where the exterior that was used to handle laboratory materials is now encased on the inside, and the clean interior is what is visible. Then you can dispose of your gloves appropriately and wash your hands. Always change your gloves immediately if you get anything on them. Note that while the hazards associated with your physics lab experiments are very low, this is still a great chance to practice the correct use and removal of gloves. Personal items, such as backpacks and coats, should be stored in the cubbies that are found in the lab. Do not leave these items out on bench tops or the floor, as they present a tripping hazard and can become contaminated or damaged. No food or drink, including water, is allowed in the lab. Before we jump into some items specific to your course, let's go over a few last general safety tips. Never enter and/or work in a lab space unless your TA or instructor is present. When you work in a lab space for the first time, identify the exit locations in the event of an emergency. Your TA should go over the evacuation plan with you on your first day in the lab. Understand the hazards of the substances you'll be working with in the lab before you work with them. One great source of information for better understanding these hazards is the SDS or safety data sheets. These are available on the web for almost all chemicals and reagents and can usually be found by searching SDS for the name of that substance in a search engine. For example, to learn more about glycerol, a substance that is used in Physics 2015, you could open a search engine and type SDS for glycerol. In the Physics 2015 and 2025 courses, each group will have a designated bin. The basic supplies held in each bin include 4 different colored whiteboard markers, an eraser, a Sharpie, a ruler, a protractor, a pair of scissors, and a rubber duck. Throughout the semester, there will be more added to the bin, depending on which lab you are working on, such as chamber well slides, cutting boards and knives, bubble levels, etc. Each of the bins should have a number which corresponds with the station you are working at. As you go through the semester, you might find that you need to replace some of the general bin supplies. The general supplies, as well as the whiteboard supplies, can be found in these 2 drawers in your lab. Throughout this course, you will be using different pieces of equipment and solutions. The equipment used includes optical microscopes, vortexers, spectrometers, fiber optic cables, calipers, DC power supplies, and gas spectral emission lamps. In the Physics 2015 lab course, the only chemicals that will be used are glycerol, water, silicon microspheres, and sodium chloride. For the Physics 2025 course, the chemicals used are silicon microspheres, water, chlorophyll, isopropanol, and fluorescein. In your lab, the chemical solutions will be stored in one of 3 ways: in a microcentrifuge tube, in a 15 ml Falcon tube, or in a cuvette. The microcentrifuge tubes will be color-coded. Solutions of the same type will be in the same colored tubes, which will in turn be stored in matching colored racks. Each rack will be labeled in the same style with information in 3 lines. The first line includes the size of the sphere and the type of material the sphere is made out of. The second line includes what type of solvent the spheres are suspended in. The third line has information about the sphere concentration. The solutions stored in the 15 milliliter Falcon tubes are saline solutions and will be labeled saline. The solutions in the cuvettes have an identifying mark on their lid to help distinguish between them. These are specifically labeled on the lid because the sides of the cuvettes must remain clear of marks. After you're done using a solution, if there is any left, return the tube to the correct rack. If the tube is empty, or there is so little solution that someone else would be unable to use it, and it is a microcentrifuge tube, throw it into the garbage can. 15 milliliter Falcon tubes should be given to a TA to refill. If you ever have to dump out remaining solutions for any reason, be sure to pour those into an appropriately labeled unwanted materials container. Ask your TA if you need one, and there is not one available. When working with solutions, sometimes you will have to work with micropipettes, which require pipette tips. After you have used the pipette tip, it's important to dispose of it in the plastic beaker found at your station. The contents of these beakers will later be transferred to larger sharps containers. Never put pipette tips into the regular trash, as doing so puts both our prep and custodial staff at risk of injury. Plastic transfer pipettes that are used during the course can be thrown in the trash. Glass microscope slides can be placed either into the dirty equipment bin on the counter or the cardboard glass disposal bin. Make sure to use this glass disposal bin only for sharps waste. Other items such as gloves, paper towels, etc. go into the normal trash cans. Chamber well slides are special types of slides. These are quite expensive and we wash and reuse them. Therefore, unless they are broken, always return chamber well slides to the dirty equipment. Remember that you can always ask your TA, instructor, or other lab personnel if you have any questions about the laboratory. Thank you for your attention and doing your part to maintain a clean and safe lab environment for everyone.